Excretory products and their elimination. Class 11 NCERT. Dr. Jayachandran Tejas. Animals accumulate wastes through metabolic activities and excess ingestion. These wastes include nitrogenous compounds such as ammonia, urea, and uric acid. Ammonia is highly toxic, is a prevalent nitrogenous waste in aquatic animals. Urea, less toxic than ammonia, is the primary nitrogenous waste in mammals. Uric acid, the least toxic, is found in the waste of birds and reptiles. Other wastes include carbon dioxide, a byproduct of cellular respiration, and water and ions, sodium, potassium, chloride, phosphate, sulfate. Ammonia stands out as the most toxic nitrogenous waste, necessitating a large amount of water for elimination. On the other hand, uric acid, the least toxic nitrogenous waste, requires only a small amount of water for excretion. 1. How do animals accumulate wastes? By metabolic activities. By excess ingestion. 2. Which of these wastes? Nitrogenous wastes: ammonia, urea, uric acid, others carbon dioxide, water, ions sodium, potassium, chloride, phosphate, sulfate. 3. Most toxic nitrogenous waste? Ammonia. 4. Least toxic nitrogenous waste? Uric acid. 5. How much water is needed to eliminate ammonia and uric acid? Ammonia large amount, uric acid small amount. The process of excreting ammonia is known as ammonotalism. Many bony fishes, aquatic amphibians, and aquatic insects are examples of ammonotelic animals. In fish, ammonia is excreted across gill surfaces through diffusion as ammonium ions, and another method involves diffusion across the body surface. Kidneys do not play a role in the removal of ammonia. The terrestrial adaptation of animals led to the production of less toxic nitrogenous wastes like urea and uric acid to conserve water, as highly toxic ammonia requires a significant amount of water for elimination. Mammals, terrestrial amphibians, and marine fishes are ureotelic animals that primarily excrete urea. In ureotelic animals, ammonia produced by metabolism is converted into urea in the liver. Urea is released into the blood by the liver, taken up by the kidneys, and then removed from the body. Some amount of urea may be retained in the kidneys to maintain osmolarity. Animals that excrete nitrogenous wastes as uric acid are termed uricotelic animals. Examples of uricotelic animals include reptiles, birds, land snails, and insects. Uricotelic animals excrete uric acid in the form of pellets or paste. 6. What is the process of excreting ammonia called? Ammonotalism. 7. Which animals are ammonotelic? Many bony fishes, aquatic amphibians, and aquatic insects. 8. How is ammonia excreted in fish? Ammonia is excreted across gill surfaces by diffusion as ammonium ions. 9. What is the other method of excreting ammonia? Diffusion across body surface. 10. What is the role of kidneys in the removal of ammonia? Kidneys do not play any role in the removal of ammonia. 11. Why did terrestrial adaptation of animals lead to the production of less toxic nitrogenous wastes like urea and uric acid? To conserve water. Because highly toxic ammonia needs lot of water for elimination. 12. Which animals are ureotelic? That is, they excrete urea. Mammals, terrestrial amphibians, and marine fishes. 13. How is urea produced in ureotelic animals? Ammonia produced by metabolism is converted into urea by the liver. 14. How is urea removed in ureotelic animals? Urea is released into blood by liver. This is taken up by kidneys and removed from the body. 15. Some amount of urea may be retained in kidneys. Why? To maintain osmolarity. 16. What is the term for animals that excrete nitrogenous wastes as uric acid? Uricotelic animals. 17. Which are the uricotelic animals? Reptiles, birds, land snails, and insects. 18. How do uricotelic animals excrete uric acid? In the form of pellets or paste. In most invertebrates, excretory structures take the form of simple tubular structures. Vertebrates, on the other hand, possess complex tubular organs known as kidneys as their primary excretory organs. Certain invertebrates, including platyhelminthes, flatworms, e.g., planaria, rotifers, some annelids, and the cephalochordate Amphioxus, feature proteinephridia or flame cells as their excretory organs. The primary function of proteinuria is ionic and fluid volume regulation, specifically osmoregulation. Other invertebrates, such as earthworms and certain annelids, utilize nephridia as their excretory structures. Nephridia serve the dual functions of removing nitrogenous wastes and osmoregulation. Malpighian tubules function as excretory organs in many insects, including cockroaches. These tubules perform the tasks of removing nitrogenous wastes and osmoregulation. Crustaceans like prawns employ antennal glands or green glands to perform their excretory functions. 19. What are the excretory structures in most invertebrates? Simple tubular forms. 20. What do vertebrates have as their excretory organs? Complex tubular organs called kidneys. 21. Which invertebrates have proteinuria or flame cells as excretory organs? Platyhelminthes, flatworms, e.g., Planaria, rotifers, some annelids, and the cephalochordate Amphioxus. 22. What is the primary function of proteinuria? Ionic and fluid volume regulation, i.e., osmoregulation. 23. Which invertebrates have nephridia as earthworms and other annelids? Excretory structures. 24. What are the functions of nephridia? Removal of nitrogenous wastes and osmoregulation. 25. Which invertebrates have Malpighian tubules as excretory organs? Most insects, including cockroaches. 26. What functions do Malpighian tubules perform? Removal of nitrogenous wastes and osmoregulation. 27. What perform the excretory function in crustaceans like prawns? Antennal glands or green glands. 19.1 Human excretory system. The human excretory system comprises a pair of kidneys, a pair of ureters, a urinary bladder, and a urethra. The kidneys, situated between the last thoracic and third lumbar vertebrae, near the dorsal inner wall of the abdominal cavity, are reddish-brown, bean-shaped structures. Adult human kidneys average 10 to 12 centimeters in length, 5 to 7 centimeters in width, and 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams. Towards the center of the inner concave surface of the kidney, there is a notch called the hilum, through which the ureter, blood vessels, and nerves enter. The broad funnel-shaped space inner to the hilum is known as the renal pelvis, with projections called calyces. The outer layer of the kidney is encapsulated in a tough capsule. Inside the kidney, two main zones exist, an outer cortex and an inner medulla. The medulla is composed of conical masses called medullary pyramids, while the extensions of the cortex between these pyramids are known as renal columns or columns of Bertini. The functional unit of the kidney is the nephron, and each kidney contains nearly 1 million nephrons. A nephron consists of two parts, the glomerulus and the renal tubule. The glomerulus is a tuft of capillaries formed by the afferent arteriole, enclosed by Bowman's capsule, collectively known as the Malpighian body or renal corpuscle. The renal tubule includes the proximal convoluted tubule, PCT, Henle's loop with descending and ascending limbs, and the distal convoluted tubule, DCT. Collecting duct is a straight tube into which the distal convoluted tubules, DCTs, of many nephrons open. Collecting ducts converge and open into the calyces of the renal pelvis through medullary pyramids. The Malpighian corpuscle, proximal convoluted tubule, PCT, and distal convoluted tubule, DCT, are located in the cortical region of the kidney. The loop of Henle extends into the medulla, with cortical nephrons having a short loop, and juxtamedullary nephrons having a long loop that runs deep into the medulla. The efferent arteriole from the glomerulus forms a network of peritubular capillaries around the renal tubule. Within the peritubular capillaries, a minute vessel running parallel to Henle's loop forms a U shape and is called the vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. 28. What are the components of the human excretory system? A pair of kidneys, a pair of ureters, a urinary bladder, and a urethra. 29. Describe the appearance and location of kidneys in humans. Kidneys are reddish-brown, bean-shaped structures situated between the levels of the last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. 30. What are the dimensions and weight of adult human kidneys? Measure 10 to 12 centimeters. An adult human kidney on average. In length, 5 to 7 centimeters in width, 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams, 31. What is the structure found towards the center of the inner concave surface of the kidney? A notch called hilum through which ureter, blood vessels, and nerves enter. 32. What is the broad funnel-shaped space inner to the hilum called? Renal pelvis with projections called calyces, singular calyx. 33. Describe the outer layer of the kidney. A tough capsule. 34. What are the two main zones inside the kidney? An outer cortex and an inner medulla. 35. Medulla is made of conical masses projecting into calyces. What are they medullary pyramids called? 36. Cortex extends between the medullary pyramids. What are these extensions called? Renal columns or columns of Bertini. 37. What is the functional unit of the kidney, and how many does each kidney have? Nephron. Each kidney has nearly 1 million nephrons. 38. What are the two parts of a nephron? The glomerulus and the renal tubule. 39. What is glomerulus? The tuft of capillaries formed by the afferent arteriole, which is a fine branch of the renal artery. Subsequently, blood from the glomerulus is carried away by an efferent arteriole. 40. What structure encloses the glomerulus? Bowman's capsule. 41. Glomerulus plus Bowman's capsule is called Malpighian body or renal corpuscle. 42. Describe the parts of the renal tubule. Proximal convoluted tubule, PCT, Henle's loop, and distal convoluted tubule, DCT. 43. What is the continuation of the tubule after the Malpighian body? Proximal convoluted tubule, PCT. 44. What is the distinctive shape of Henle's loop, and what are its two parts? Hairpin-shaped, descending and ascending limb. 45. What follows the ascending limb of the loop of Henle? Distal convoluted tubule, DCT. 46. What is the straight tube into which the collecting duct? Distal convoluted tubules, DCTs, of many nephrons open. 47. Where do collecting ducts converge and open into? Through medullary pyramids into the calyces into renal pelvis. 48. What is the location of the Malpighian corpuscle, proximal convoluted tubule, PCT, and distal convoluted tubule, DCT, within the kidney? Cortical region of the kidney. 49. Into which kidney region does the loop of Henle extend, and what is its typical length of loop of Henle in the majority of nephrons? Dips into the medulla. It is usually short and extends only a little into the medulla. 50. What term is used for nephrons with a cortical nephrons? Short loop of Henle that extends minimally into the medulla. 51. Some nephrons have very long loop of Henle that runs deep into the medulla. What are these nephrons called? Juxtamedullary nephrons. 52. What does the efferent arteriole from the glomerulus form around the renal tubule? A network of peritubular capillaries. 53. What is the minute vessel? Within the peritubular capillaries, running parallel to Henle's loop, forming a U shape. Vasa recta. 54. In which type of nephrons is the vasa recta absent or highly reduced? Cortical nephrons. 19.2 Urine formation. Urine formation involves three main processes: glomerular filtration, tubular reabsorption, and tubular secretion. The part of the nephron responsible for the first step of blood filtration in urine formation is the glomerulus. The term for the filtration of blood carried out by the glomerulus is glomerular filtration. On average, the kidneys filter 1,100 to 1,200 milliliters of blood per minute. Roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute is filtered by the kidneys. The filtration of blood through the glomerulus is caused by glomerular capillary blood pressure. During glomerular filtration, blood is filtered through three layers: the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The cells of the epithelium of Bowman's capsule are called podocytes. The minute spaces between podocytes are referred to as filtration slits or slit pores. The process where almost all constituents of the plasma, except proteins, pass into the lumen of Bowman's capsule is termed ultrafiltration. The amount of filtrate formed by the kidneys per minute is known as glomerular filtration rate, GFR. In a healthy individual, the GFR is approximately 125 milliliters per minute, equivalent to 180 liters per day. Juxtaglomerular apparatus, JGA, is a special sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole at their contact point. The role of JGA in the regulation of GFR is that a fall in GFR can activate JG cells to release renin, stimulating glomerular blood flow and restoring GFR to normal. Nearly 99% of the glomerular filtrate is reabsorbed by the renal tubules. In a day, 1.5 liters of urine is formed from 180 liters of glomerular filtrate. The process where water and other substances are reabsorbed by the renal tubules is called reabsorption. The reabsorption of nitrogenous wastes, such as urea, in the renal tubule is accomplished by passive transport. The reabsorption of glucose, amino acids, and Na+ in the renal tubule is accomplished by active transport. The reabsorption of water in the initial segments of the renal tubule is accomplished passively. The process where tubular cells secrete substances like H+, K+, and ammonia into the filtrate during urine formation is called tubular secretion. The function of tubular secretion is the maintenance of ionic and acid-base balance of body fluids. 55. What are the three main processes involved in urine formation? Glomerular filtration, tubular reabsorption, and tubular secretion. 56. Which part of the nephron is responsible for the filtration of blood in the first step of urine formation? Glomerulus. 57. What is the term for the filtration of blood carried out by the glomerulus? Glomerular filtration. 58. How much blood is filtered by the kidneys per minute? 1,100 to 1,200 milliliters. 59. What fraction of the blood pumped out by each ventricle of the heart is filtered by the kidneys in a minute? 1/5. 60. What causes the filtration of blood through the glomerulus? Glomerular capillary blood pressure. 61. Name the three layers through which blood is filtered during glomerular filtration. Endothelium of glomerular blood vessels, epithelium of Bowman's capsule, and basement membrane between above two layers. 62. What are cells of epithelium of Bowman's capsule called? Podocytes. 63. What are the minute spaces between podocytes called? Filtration slits or slit pores. 64. What is the term for the process where almost all constituents of the plasma, except proteins, pass into the lumen of Bowman's capsule? Ultrafiltration. 65. What is the term for the amount of filtrate formed by the kidneys per minute? Glomerular filtration rate, GFR. 66. What is the GFR in a healthy individual? 125 milliliters per minute, equivalent to 180 liters per day. 67. What is JGA? It is a special sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole at their contact point. 68. What is the role of the juxtaglomerular apparatus, JGA, in the regulation of GFR? When there is a fall in GFR, JG cells release renin, increasing glomerular blood flow and restoring GFR to normal. 69. What percentage of the glomerular filtrate is reabsorbed by the renal tubules? 99%. 70. In a day, how much urine is formed from how much glomerular filtrate? 1.5 L urine. 180 L glomerular filtrate. 71. What is the process called where water and other substances are reabsorbed by the renal tubules? Reabsorption. 72. How is the reabsorption of nitrogenous wastes, such as urea, accomplished in the bypassive transport? Renal tubule. 73. How is the reabsorption of glucose, amino acids, Na+ accomplished in the renal tubule? By active transport. 74. How is the reabsorption of water in the initial segments of the renal tubule accomplished? Passively. 75. What is the term for the process where tubular cells secrete substances like H+, K+, and ammonia into the filtrate during urine formation? Tubular secretion. 76. What is the function of tubular secretion? Maintenance of ionic and acid-base balance of body fluids. 19.3 Functions of the tubules. The proximal convoluted tubule, PCT, is characterized by a lining of simple cuboidal brush border epithelium, serving the purpose of increasing the surface area for reabsorption. This segment plays a pivotal role in the reabsorption process, reclaiming nearly all essential nutrients and 70 to 80% of electrolytes and water. Additionally, The PCT contributes to the maintenance of pH and ionic balance in body fluids through the selective secretion of hydrogen ions, ammonia, and potassium ions into the filtrate. Simultaneously, it absorbs bicarbonate, HCO3, from the filtrate. Moving further along the nephron, the descending limb of Henle's loop exhibits permeability to water while remaining impermeable to electrolytes. As the filtrate progresses down this limb, it undergoes concentration due to the water permeability. In contrast, the ascending limb of Henle's loop is impermeable to water but actively or passively. Transports electrolytes. As the filtrate ascends, it becomes diluted due to the passage of electrolytes to the medullary fluid. Notably, reabsorption is at a minimum in the ascending limb. The distal convoluted tubule, DCT, is responsible for conditional reabsorption of sodium, Na+, and water, reabsorption of bicarbonate, HCO3, and the secretion of hydrogen ions, potassium ions, and ammonia, NH3. Its primary function is to contribute to the maintenance of pH and sodium-potassium balance in the blood. The collecting duct, extending from the cortex to the inner parts of the medulla, is crucial for further urine concentration. It facilitates the reabsorption of large amounts of water, allowing the production of concentrated urine. Additionally, the collecting duct contributes to the maintenance of osmolarity in the medullary interstitium by permitting the passage of small amounts of urea from the filtrate into the medullary interstitium. It selectively secretes H+ ions to maintain the pH of blood and K+ ions fall. The ionic balance. The key feature enabling mammals to produce a concentrated urine lies in the ability of Henle's loop and vasa recta to establish a counter-current system. The flow of filtrate in opposite directions in the limbs of Henle's loop, forming a countercurrent, and the participation of vasa recta in this pattern contribute to increasing osmolarity towards the inner medullary interstitium, reaching up to 1200 mosmol/L. The main contributors to this osmolarity gradient are sodium chloride and urea. Sodium chloride is transported out from the ascending limb of Henle's loop, exchanged with the descending limb of vasa recta, and returned to the interstitium by the ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of the ascending limb of Henle's loop, transported back to the interstitium by the collecting tubule. The overall term for the transport of substances facilitated by this countercurrent arrangement is the countercurrent mechanism. This mechanism helps maintain a concentration gradient in the medullary interstitium. The presence of such a gradient allows for an easy passage of water from the collecting tubule to the interstitium, thereby concentrating the filtrate, urine. Human kidneys demonstrate a remarkable capability in urine concentration, producing urine nearly 4 times more concentrated than the initial filtrate formed. 77. What type of epithelium lines the proximal convoluted tubule, PCT? Simple cuboidal brush border epithelium. 78. Why does the PCT have a brush border epithelium? To increase the surface area for reabsorption. 79. What percentage of essential nutrients, electrolytes, and water are reabsorbed by the PCT? Nearly all essential nutrients, and 70 to 80% of electrolytes and water. 80. How does the PCT contribute to secreting hydrogen ions, ammonia, and maintaining the pH and ionic balance of body fluids? Potassium ions into filtrate, absorbing HCO3 from filtrate. 81. Describe the permeability of the descending limb of Henle's loop. Permeable to water but impermeable to electrolytes. 82. What happens to the filtrate as it moves down the descending limb of Henle's loop? It gets concentrated due to the permeability to water. 83. What is the permeability of the ascending limb of Henle's loop to water and electrolytes? Impermeable to water but allows transport of electrolytes actively or passively. 84. What happens to the filtrate as it moves up the ascending limb of Henle's loop? It gets diluted due to passage of electrolytes from filtrate to the medullary fluid. 85. In which limb of Henle's loop is reabsorption minimum? The ascending limb. 86. What role does the ascending limb of Henle's loop play in maintaining medullary interstitial fluid osmolarity? It contributes to the maintenance of high osmolarity of medullary interstitial fluid. 87. What are the events which happen in the distal convoluted tubule, DCT? Conditional reabsorption of Na+ and water. Reabsorption of HCO3. Secretion of hydrogen ions, potassium ions, and NH3. 88. What is the function of the DCT? Maintenance of pH and sodium-potassium balance in blood. 89. What is the anatomical location of the collecting duct? The collecting duct extends from the cortex of the kidney to the inner parts of the medulla. 90. What is the primary function of the collecting duct regarding water reabsorption? Large amounts of water can be reabsorbed from the collecting duct to produce a concentrated urine. 91. How does the collecting duct contribute to the maintenance of osmolarity in the medullary interstitium? By allowing the passage of small amounts of urea from filtrate into the medullary interstitium. 92. What role does the collecting duct play in maintaining the pH of blood? The collecting duct selectively secretes H+ ions. 93. How does the collecting duct contribute to the ionic balance of blood? The collecting duct selectively secretes K+ ions. 94. What is the key feature that enables mammals to produce a concentrated urine? The ability of the Henle's loop and vasa recta to establish a countercurrent system. 95. What is the flow pattern of filtrate in the limbs of Henle's loop? The flow of filtrate is in opposite directions, forming a countercurrent. 96. In addition to Henle's loop, what else participates in the countercurrent pattern? The vasa recta, the blood vessel associated with Henle's loop, also has a countercurrent pattern. 97. What is the significance of the countercurrent arrangement in Henle's loop and vasa recta? It produces increasing osmolarity towards the inner medullary interstitium, from 300 mosmol/L in the cortex to 1200 mosmol/L in the inner medulla. 98. What substances contribute to the gradient in osmolarity in the medullary interstitium? Sodium chloride and urea. 99. How is sodium chloride transported in the countercurrent mechanism? Sodium chloride is transported out from the ascending limb of Henle's loop. This enters descending limb of vasa recta. This is returned to the interstitium by the ascending portion of vasa recta. 1. How does urea move in the countercurrent mechanism? Small amounts of urea enter the thin segment of the ascending limb of Henle's loop from the interstitium. This is transported back to the interstitium by the collecting tubule. 2. What is the overall term for the transport of substances facilitated by the countercurrent arrangement? Countercurrent mechanism. 3. How does countercurrent mechanism help in concentrating urine? Countercurrent mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such a gradient helps in an easy passage of water from the collecting tubule to the interstitium, thereby concentrating the filtrate, urine. 4. What is the remarkable capability of human kidneys regarding urine concentration? Human kidneys can produce urine nearly 4 times more concentrated than the initial filtrate formed. 19.5 Regulation of kidney function. The functioning of the kidneys is efficiently monitored and regulated by hormonal feedback mechanisms involving the hypothalamus, juxtaglomerular apparatus, JGA, and to a certain extent, the heart. Hypothalamus osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. In response to an excessive loss of fluid from the body, these osmoreceptors stimulate the hypothalamus to release antidiuretic hormone, ADH, or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from the latter parts of the tubule, preventing diuresis. An increase in body fluid volume switches off the osmoreceptors, suppressing ADH release to complete the feedback loop. ADH not only prevents diuresis but also constricts blood vessels, leading to increased blood pressure. This increase in blood pressure, in turn, can enhance the glomerular blood flow and the glomerular filtration rate, GFR. The juxtaglomerular apparatus, JGA, plays a complex regulatory role in kidney function. A decrease in glomerular blood flow, blood pressure, or GFR can activate JG cells to release renin. Renin, in turn, converts angiotensinogen in the blood to angiotensin I and further to angiotensin II. Angiotensin II, a potent vasoconstrictor, increases glomerular blood pressure, leading to an elevation in GFR. Additionally, angiotensin II activates the adrenal cortex to release aldosterone, promoting the reabsorption of Na+ and water from the distal parts of the tubule, further increasing blood pressure and GFR. This intricate mechanism is commonly known as the renin-angiotensin mechanism. In the event of an increase in blood flow to the atria of the heart, atrial natriuretic factor, ANF, is released. ANF causes vasodilation, dilation of blood vessels, resulting in reduced blood pressure. The ANF mechanism acts as a check on the renin-angiotensin mechanism, providing a counterbalancing influence. 5. How are the kidneys efficiently monitored and regulated in their functioning? Hormonal feedback mechanisms involving the hypothalamus, juxtaglomerular apparatus, JGA, and the heart. 6. What activates osmoreceptors in the body? Changes in blood volume, body fluid volume, and ionic concentration. 7. What is the response of osmoreceptors to an excessive loss of fluid from the body? Osmoreceptors stimulate the hypothalamus, leading to release antidiuretic hormone, ADH, or vasopressin from the neurohypophysis. ADH leads to water reabsorption from latter parts of tubule, leading to increase in blood volume. ADH constricts blood vessels, leading to increased BP. 8. How is the release of ADH regulated in response to an increase in body fluid volume? Increase in body fluid volume switches off the osmoreceptors, thus suppressing the release of ADH, completing the feedback loop. 9. What is the effect of ADH on diuresis? Prevents diuresis. 10. What is the effect of ADH on kidney function? Increase in blood pressure increases the glomerular blood flow and thereby the GFR. 11. What is the role of the juxtaglomerular apparatus, JGA, in kidney function regulation? A decrease in glomerular blood flow, blood pressure, or glomerular filtration. Rate, GFR, activates JG cells to release renin. Renin converts angiotensinogen in the blood to angiotensin I, and further to angiotensin II. Angiotensin II, a potent vasoconstrictor, increases glomerular blood pressure, leading to an elevation in GFR. Angiotensin II also activates the adrenal cortex to release aldosterone, which causes the reabsorption of Na+ and water from the distal parts of the tubule, further increasing blood pressure and GFR. This complex mechanism is known as the renin-angiotensin mechanism. 12. How does an increase in blood flow to the atria of the heart impact blood pressure, and what factor is released in response? An increase in blood flow to the atria of the heart causes the release of atrial natriuretic factor, ANF. ANF causes vasodilation, dilation of blood vessels, reducing blood pressure. The ANF mechanism acts as a check on the renin-angiotensin mechanism, providing a counterbalancing influence. 19.6 Micturition. Urine formed by the nephrons is carried to the urinary bladder. The initiation of urine release from the bladder is a controlled process involving stretch receptors located in the bladder walls. These receptors respond to the stretching of the bladder caused by the accumulation of urine. Upon activation, the stretch receptors send signals to the central nervous system, CNS. In response to these signals, the CNS issues voluntary motor messages that coordinate the contraction of smooth muscles within the bladder. Simultaneously, the urethral sphincter undergoes relaxation, leading to the release of urine. This process is termed micturition, and the neural mechanism governing it is known as the micturition reflex. On average, an adult human excretes approximately 1 to 1.5 liters of urine daily. The characteristics of urine include being a light yellow-colored, watery fluid with a slightly acidic nature and a pH of 6.0. The daily excretion of urea averages between 25 to 30 grams. The analysis of urine plays a crucial role in clinical diagnosis, providing insights into metabolic disorders and kidney malfunctions. For instance, the presence of glucose, glycosuria, and ketone bodies, ketonuria, in urine can serve as indicators of conditions such as diabetes mellitus. 13. What happens to urine formed by the nephrons? Urine formed by the nephrons is carried to the urinary bladder. 14. How is the release of urine initiated from the urinary bladder? Stretch receptors in the walls of the bladder send signals to the CNS in response to stretching of the bladder when it is filled with urine. The CNS sends voluntary motor messages that lead to the contraction of smooth muscles in the bladder and the simultaneous relaxation of the urethral sphincter, resulting in the release of urine. 15. What is the term for the process of micturition? Releasing urine? 16. What is the neural mechanism causing the release of urine known as micturition reflex? 17. How much urine does an average adult human excrete per day? 1 to 1.5 L18. What are the characteristics of urine in terms of color and acidity? Urine is a light yellow-colored watery fluid, slightly acidic with a pH of 6.0. 19. How much urea is excreted on average per day? 25 to 30 gm. 20. How can the analysis of urine contribute to clinical diagnosis? Analysis of urine helps in diagnosing metabolic disorders and kidney malfunctions. For example, the presence of glucose, glycosuria, and ketone bodies. Ketonuria can indicate diabetes mellitus. 19.7 Role of other organ systems in excretion. Various organs contribute to the elimination of excretory wastes, not limited to the kidneys. The lungs, liver, and skin play essential roles in this process. Lungs contribute by removing approximately 200 milliliters of CO2 every minute and significant amounts of water. The liver, the largest gland in the body, secretes bile-containing substances such as bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. These secretions are eliminated along with digestive wastes. Sweat glands in the skin eliminate substances like sodium chloride, small amounts of urea, and lactic acid, producing a watery fluid known as sweat. While the primary function of sweat is to facilitate cooling, it also aids in removing some waste products. Sebaceous glands in the skin eliminate sterols, hydrocarbons, and waxes through the secretion of sebum, providing a protective oily covering for the skin. Additionally, small amounts of nitrogenous wastes can be eliminated through saliva. 21. Besides the kidneys, which organs contribute to the elimination of excretory wastes? Lungs, liver, and skin also play a role in waste elimination. 22. How do lungs contribute to waste elimination? Lungs remove 200 milliliters per minute of CO2 and significant amounts of water daily. 23. What substances does the liver secrete, and how are they eliminated? The liver secretes bile containing bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. These substances are eliminated with digestive wastes. 24. Which is the largest gland in the body? The liver. 25. What do sweat glands in the skin eliminate, and what is the consistency of sweat glands eliminate sodium chloride and small? Sweat, amounts of urea, lactic acid, etc. Sweat is a watery fluid. 26. What is the primary function of sweat? Cooling. 27. What substances are eliminated by sebaceous glands in the skin, and what is the purpose of their secretion? Sebaceous glands eliminate sterols, hydrocarbons, and waxes through sebum, providing a protective oily covering for the skin. 28. Can nitrogenous wastes be eliminated through saliva? Yes, small amounts of nitrogenous wastes can be eliminated through saliva.